So hi everyone, and welcome to the last seminar for this academic year. It's a great pleasure to welcome Dr. Meaghan Perdue as our speaker. Meaghan is an honorary Killam postdoctoral fellow in the Developmental Neuroimaging Lab at the University of Calgary. That's with Catherine Lebel, who many of you may know. She was here at UCLA many years ago. So the Killam Fellowship has quite a competitive selection process and is awarded via the Killam Trust, which is one of Canada's largest and most prestigious endowments for scholarly activities. Meaghan completed her PhD from the University of Connecticut in 21 in developmental psychology with a specialization in the neurobiology of language under the supervision of Dr. Nicole Landy. So in her doctoral research, Meaghan investigated structural, functional, and chemical correlates of reading ability in children and applied neuroimaging genetics type approaches to study gene, brain, and behavior relationships. Meaghan has also been a recipient of the NSF Graduate Research Fellowship and is an NSF IGERT Fellow. So her current research is focused on understanding the neurobiological mechanisms behind reading and language abilities across development. And she does that using multimodal and quantitative neuroimaging techniques. So her talk today is about brain metabolites and the role they play in brain development. So MRS, as most of you know, is an inherently challenging modality. And we've collected our fair share of MRS data in children here, although we're not focused on reading and language. So we're interested to hear your findings. So let's welcome Meaghan.
Thank you. Let's make sure I can get these slides up. So thank you for inviting me today, and I'm excited to share with you some of my ongoing work. I'll also touch upon a bit of my dissertation work where I got started looking at neurochemistry in the first place, but focusing generally today on metabolites in healthy brain development, and as well as some of those connections with cognition and reading. I'd like to start just by acknowledging the team that I work with at the University of Calgary, especially Catherine Lebel, my primary supervisor, as well as our collaborators in Dr. Ashley Harris's lab, as well as her postdocs, Marilena DeMaio and Tiffany Bell, who have been a great help with developing all of this work, as well as thanking my former lab at the University of Connecticut, led by Dr. Nicole Landy, which got me started on this line of research. So I'll just start with a brief introduction to brain metabolites and MRS. Brain metabolites are involved in many aspects of brain functioning and development. They're these naturally occurring chemicals in the brain that help with the synthesis and maintenance of brain tissue. They regulate brain function, serve as neurotransmitters, control the energy supply, and this list goes on. These metabolites can change with development and aging, as well as in cases of disease and in injury. And again, this list goes on in terms of neurodevelopmental conditions. And because each metabolite can play many roles in the brain, it can be difficult to interpret and understand the effects and mechanisms of metabolites and their changes. And so this is what drives my current research. So in our MRS sequences, when we use our proton MRS at 3T, there are 5 main families of metabolites that we measure. And so these include N-acetylaspartate and N-acetylaspartyl glutamate, which is our total NAA signal, as well as our total creatine signal, which is creatine and phosphocreatine. Our choline signal is consisting of choline, but more importantly, choline-containing compounds, including phosphocholine and glycerophosphocholene, as well as myoinositol and In our research, we look at glutamate and glutamine as a combined signal, which is the most reliable and robust in our data acquisitions. And that's noted as GLX throughout my slides. So NAA is involved in neuronal metabolism, the synthesis of myelin, and the synthesis of other neurotransmitters, as well as maintaining water balance in the brain. Creatine is mostly involved in the energy homeostasis in the brain. Choline is looked at as a marker of membrane turnover, including myelin, and choline also is a precursor to acetylcholine. Myoinositol is involved in cellular signaling and lipid synthesis and is often considered a glial marker. And glutamate is our primary excitatory neurotransmitter, but it also has many roles in metabolism and the synthesis and cycling with other neurotransmitters, such as GABA. So as you can see, there are many different systems that these are involved in, and this leads me to the three main questions that I'll be touching on today. So first, do metabolite levels change across development? So I'll cover some recent work that I've done examining the patterns of development across childhood. Second is, what can metabolites tell us about tissue structure? And here I'm primarily interested in looking at white matter structure. And then finally, are metabolites related to cognitive traits and how? So prior evidence has shown that metabolite levels do show some changes, but that evidence has mainly included cross-sectional data and has shown these most dramatic changes in metabolite levels even in the first 3 months postnatally, and then continuing into the first couple of years of life. But in my work, I've really wanted to focus on this more understudied age range in early to middle childhood, where in these prior cross-sectional studies, we see relative stability, but we haven't really had research that have probed this age range longitudinally. So in our data, we've used single voxel MRS to measure metabolites in the anterior cingulate cortex and in the left temporoparietal region. And so this data was acquired from an accelerated longitudinal study. So most children in the study had one time point where we acquired data from this ACC voxel. And then in their subsequent visits, they had their MRS acquired from this left temporoparietal voxel. And that has resulted in our ACC data being primarily cross-sectional and our LTP data being longitudinal, with many subjects having 3 or more time points collected annually from the LTP. And so we examined the changes over time in these metabolites in these two brain regions. And for NAA, we found overall increases across childhood from ages 2 to 12 years. And this is fairly consistent with prior evidence that has shown ongoing increases in NAA. These age-related increases could be related to various aspects of axon development, and we think this is probably the primary driver in this age range that we're looking at, with elongation of axons, increasing axon diameter, and packing of axons, and possibly with also contributions from myelination. But this could also reflect some increases in metabolic activity, and the contributions from the NAAG signal that we measure with total NAA could relate to regulations of complex cognitive functioning, though that is speculative since NAAG only is a small part of the NAA signal that we measure. For choline, we see decreases across childhood, again, in both of these voxels. And when we looked at the quadratic effects in our longitudinal data set, so this model shown here in this rightmost panel, looked at kids with 3 or more time points of measurement for choline in the LTP, we found that there was a significant quadratic effect with more rapid decreases earlier on and a minimum point reached right around 9 years old. And so that pattern of timing likely corresponds to potential ongoing changes in white matter structure and myelination. So during myelination, choline-containing compounds are incorporated into myelin phospholipids and then no longer visible to us in our MRS signal. Though we think that the bulk of myelination probably happened before our age range, which leads us to believe that these ongoing choline changes could be related to other aspects of membrane turnover and changes in cell density that are associated with the wiring and building more efficient white matter tracts, which we know those changes continue going on through childhood, evidenced by diffusion MRI research. So for creatine, we saw increases over childhood, but this effect was only present in the temporoparietal voxel, not the ACC, and the effect was quite modest. So there's a couple possibilities. I hesitate to make conclusions that this is a region-specific effect just because we had more power to detect this effect in this left temporoparietal region than we did in the ACC, given the way our data was collected. But what we do see is this modest increase in the LTP, which could reflect increasing energy demands, as well as increasing local synaptic activity. And this is interesting in relation to prior evidence from our lab in an earlier study from this same sample of children, where my colleagues found evidence of increasing resting state local activity and global connectivity in over early childhood linked to an LTP region. I believe the inferior parietal lobule showed these increasing patterns of activity, which could be related to these creatine changes that we're observing in this region. We didn't see any significant changes in myoinositol over childhood. And when we look back into the literature, this is consistent with observations of stability in myoinositol after early development. So there are studies using MRS in utero and in preterm born infants that show rapid changes in prenatal and in early postnatal development up until around 3 months old. And then subsequently, there's stabilization of myoinositol. So we are likely capturing that period of stability here. And then finally, for our glutamate and glutamine signal, we didn't see any significant linear effects looking at either voxel, but when we applied our quadratic models, we found that there was a significant quadratic fit in our longitudinal data with an early increase in glutamate followed by a later decrease. And this could reflect changes in metabolism and in the balance of excitatory and inhibitory neural activity, as well as in neural synchronization. And we likely think that this is just part of the developmental tuning of excitability in networks that support cognition and language, such as the networks that the LTP is involved in. And what we see as well is that in studies that look at adolescents, they do tend to see linear decreases in GLX across adolescence. So we seem to be capturing perhaps the beginning of that downturn after an early increase in GLX. So this, I think, merits some further investigation, especially if I go back here. although we have this overall fit of this inverted U shape, there does seem to be a lot of inter-individual variability in both the shapes of these curves and the timings of their peaks in GLX. So this is something I'm interested in trying to pick apart a bit further and figure out if it's related to any other traits in these children across development. So far, we do find that metabolites change across childhood, though as I note, there's a need for more research to examine different brain regions, to expand this age range further into adolescence, and then also, earlier into infancy, and link these changes in metabolites to changes in brain structure, function, and behavior, all that we know are changing throughout childhood as kids develop. So, as I mentioned, there are a lot of different interpretations for what's going on with these metabolite changes across childhood, and it can be difficult to interpret in both healthy brains and in cases of disorders and disease or injury, exactly what the mechanisms are that are driving these changes or what these might reflect about our underlying brain tissue. So this is what leads me to my next line of research and kind of what I'm digging into right now. And I'm interested in what these metabolites tell us about the underlying tissue structure. So for these studies, I am focusing mainly on the total NAA signal and the total choline signal, because these are the two metabolites that have been the most consistently associated with tissue structure, with myelin, and with membranes. So there are various possibilities in what these levels of NAA and choline indicate about the brain, and they could be related to myelin turnover, which is something that's seen a lot in demyelinating disorders, such as multiple sclerosis. Some animal models show that during periods of demyelination, there are increases in choline levels, particularly that are seen, and this is likely because of the GPC part of the choline signal occurs as a product of myelin breakdown. So we can see these fluctuations as myelin is broken down and rebuilt. The NAA and choline can also indicate aspects of axon packing and kind of the density of axons in a fiber bundle, as well as the axon diameter. Especially, I think NAA is most likely related to axon diameter, since when we measure NAA, we're measuring the NAA typically in cell bodies and axons, but not in myelin. So a larger axon diameter could be home to more NAA for us to measure with MRS. And then finally, these metabolites could be related to cell density. So based on their association with membranes, if we're packing, more membranes and more cells into a space, this could show up as higher or lower levels of NAA and choline. So one model that has been proposed to link NAA and choline to white matter integrity suggests that reduced levels of NAA paired with elevated levels of choline are associated with reduced white matter integrity. And so this 2014 paper was specifically looking at some children and adolescents, I believe, who were exposed to risk for addictions. And they proposed that possibly lower levels of NAA inside axons would mean there's reduced NAA available for myelin synthesis, and that elevated levels of choline in our glial cells would be associated with higher rates of myelin turnover, or that ongoing breakdown and rebuilding of the myelin sheath. And that would be associated with lower white matter integrity, usually measured in diffusion imaging with FA. So in my work, I've mainly wanted to disentangle this a little bit. And since this model really proposes that the link here has to do with myelin, I wanted to look at a myelin-specific measure of brain tissue rather than just looking at DTI data. The previous work looking at DTI had shown that there are some positive associations between NAA and fractional anisotropy measures, but they hadn't really found anything related to choline, and the work is very limited overall. I was actually a bit surprised to see. So in these studies, I wanted to look at myelin-specific measures as well as more advanced models of diffusion MRI to try to understand what properties of the white matter tissue are reflected in our MRS measures. So in this study, we've measured myelination with quantitative inhomogeneous magnetization transfer imaging, or our QIHMT measure, which has been shown to be both sensitive and specific to myelin content. So here I'm showing our QIHMT map, and you can see that there is quite high contrast between our white matter and gray matter areas. And this kind of shows our specificity to myelin. And this was collected in the same children that participated in the longitudinal study, looking at the metabolite development. And for this first analysis, I've just looked across the board. Is myelination related to choline and NAA in these kids in our left temporoparietal voxel? So looking just within this voxel region, I took the average QIHMT measure from the same region where the children's NAA and choline measurements were taken. And we found that actually for both NAA and choline, there are positive associations with myelination. So this is consistent with the model and what we would predict for NAA, that higher levels of NAA indicate high availability for synthesis of myelin, and this positive relationship. However, the effect for choline was actually the opposite direction than we had predicted. We thought that the higher levels of choline would be associated with more turnover and breakdown products of myelin, rather than more myelin or more myelin synthesis. So it's kind of leading us to question whether these models that are really based on disease states and things like multiple sclerosis are the appropriate ways to interpret these metabolite effects in healthy brains, and especially in healthy child brains. So what we're thinking here is that choline is just associated overall with membrane content, including myelin, and that the elevated choline could be associated with some restructuring of the axon tissues and axon bundles without necessarily breaking down the overall myelin content signal. And our overall myelin content here is not related to the tract integrity and, how well these fibers connect with one another. So we wanted to add another layer to this and understand how these relationships might show up with the diffusion MRI measures that we can get to understand the tissue structure in a different way. So we are going to be doing that with this data set as our next step, looking at measures of fiber density and fiber bundle integrity, as well as the neurite dispersion, the NODI model. But I do have preliminary evidence from my PhD work. So during my dissertation work, I had started to probe these relationships, looking at multi-shell diffusion-weighted MRI and using pixel-based analysis performed with MR-Trix in a left temporoparietal voxel. So again, this is a different data set, but a similar acquisition and are a smaller sample. So these were in 8 to 12-year-old children. And using our multi-shell diffusion MRI, we did the pixel-based analysis, which can give us measures of fiber density and fiber cross-section, and this combined fiber density and cross-section metric. So compared to a normal fiber bundle, reduced fiber density just shows that we have reduced axon packing within the bundle. Reduced fiber cross-section is kind of as you would expect, a thinner and smaller axon bundle. And we can also see a case where there is both reduced cross-section and reduced packing of the axons, which would show up in this way. So get my slides to advance. What we found in this study from my dissertation, and this is still preliminary evidence, it was just 27 kids, but we did find that lower choline levels, and here they were measured in reference to creatine, were associated with more efficient white matter fiber structure. So there was this negative correlation between choline and our fiber density and cross-section metric. So having lower levels of choline meant you had stronger and more efficient white matter bundles in the fibers that passed through this left temporoparietal region. And in contrast, we see that for NAA, the effect was positive. So higher levels of NAA in this region were associated with a higher fiber density and cross-section metric, and thus more efficient white matter fiber structure passing through this voxel. So here, I'm really curious to unpack this a bit more and why we're seeing in the other data set, my Calgary data, that myelination is positively associated with choline measures, whereas here we're seeing that the fiber structure and this cross-section and density of our white matter fibers is negatively associated with choline. So I'll be continuing this work to try to see if these similar effects show up when we look at the diffusion data in our Calgary cohort. So what I've come to so far is that NAA and choline are related both to myelin content and to white matter fiber structure, but we have a lot of work to unpack this and understand really what the tissue properties are that are related to these measurements of metabolites to understand this further. So the final aspect of my work that I'll touch on today is relating metabolite levels to cognitive traits and specifically looking at reading ability. So there have been a number of studies now looking at autism and ADHD and dyslexia and trying to understand differences in the metabolite levels and show that they're related to various aspects of these traits and of aspects of cognition. And looking at reading specifically, there's been studies showing that elevated choline and glutamate levels are associated with reading disability, and then other studies that have shown reduced NAA in reading disability. And so there's mixed effects overall. As I'm sure many of you know who have done any MRS work, there are a lot of methodological differences in the way that you can collect the data and quantify metabolites. So we really just need more evidence to understand and see which effects turn out to be the most consistent. But there is fairly consistent evidence linking choline to reading abilities. So again, going back to my dissertation work, I investigated whether choline and NAA are related to reading ability in our sample of 8 to 12-year-old children with and without dyslexia. And we found some preliminary evidence. Again, these are around 27 kids in these samples. So it's a small data set, but hopefully leading to more interesting findings, showing that there is a negative association between choline and reading ability in our data. And there's a positive association between NAA and reading ability in this data set, again, measured in a left temporoparietal region. So it does appear that in this region that we know is important for reading, for orthographic phonological integration, there do seem to be metabolite effects associated with reading ability. And this is also overlapping in a region that has often shown differences using DTI in the white matter integrity and its association with reading ability and dyslexia. So again, I'm hoping to unpack this a bit further. And so we do have some evidence that metabolites are related to cognitive traits, but we do need to do more work to understand the nature of this relationship. So to put it all together, going back to this interpretation of the associated tissue structure underlying differences in NAA and choline measurements from MRS. And our initial models and what has driven a lot of my work previously was this idea that came from, again, disease states like multiple sclerosis, that elevated choline and reduced NAA is related to myelin turnover and this ongoing breakdown and rebuilding of the myelin sheath. However, my evidence actually shows that choline was positively associated with myelin content in our healthy developing children. So I'm not sure that this interpretation based on a diseased brain state is the best to guide our understanding of what's going on in our healthy child brains. And so I'd like to focus more going forward on these other aspects of the white matter fiber structure, as well as even looking at cortical fiber structure to understand how factors such as the axon diameter and axon packing, as well as the organization of the fibers, both within deep white matter as well as in the cortex, could be related to cognitive traits such as reading ability. So as I go forward, I'm hoping to dig into these aspects a bit more using our advanced diffusion modeling and linking it again back to both the myelin content as well as the tissue structure organization to see if we can draw the links between our metabolites and cognitive traits based on the underlying tissue structure. Another area that I'd like to go with this is to dig in more to our multimodal data in a more sophisticated way. So for now, I've done a lot of regression analysis and correlations to identify kind of these bivariate associations between tissue structure and metabolites or metabolites and behavior. And I think we have an opportunity here and a need here to put these all together and understand how these metabolites and tissues and even aspects of brain function all come together to be associated with a given trait such as reading ability. And so two kind of directions I see as possible for this will be to look at whether we have latent brain metabolite profiles. So are there different groups that emerge from our data who maybe have higher levels of choline and lower levels of NAA and typical levels of creatine versus kids who might have higher levels of GLX but typical levels of everything else? And do these then map on to different cognitive traits or neurodevelopmental profiles? Another direction that I see for this as well is to identify how our metabolites fit into other aspects of our brain structure and function, and to use some independent component analysis or other kind of factor analysis methods to identify whether, putting data together from different modalities, including our myelination measures, our diffusion measures, and our metabolite measures, can tell us something about factors that are associated kind of as a whole with white matter integrity, or with aspects of function and excitation, neural noise, and then do these factors then relate more strongly to cognitive traits than any single metric alone. So these are things that, maybe I can return and update you on sometime in the future, and I hope will result in some exciting findings as well. And I thank you for your attention, and I think we should have time for questions.
Great talk, Meaghan. Thanks very much. So many questions. So in the sample that you had, were there any ADHD traits underlying your population?
Yes. So our Calgary sample, make sure I can see people. So for the Calgary sample, we have kids that were just recruited pretty early on before any diagnosis for typical development, but a few of them might go on to have some diagnoses, though it's not something I controlled for in that analysis. It's a very small number in the sample with things like ADHD, dyslexia, kind of learning disabilities, but any more severe conditions or neurodevelopmental disorders that came up were excluded from the study. So yes, there could be, and that's something that I don't think there's quite enough kids in our sample with ADHD to actually tease apart. But it would be interesting, especially with that ACC data.
Right. So the reason I asked is we also, so I was not leading that study. My colleagues, we were looking at ADHD and prenatal alcohol exposure.
Okay.
And we collected some MRS data in these kids. And I noticed that, so I know you were actually not looking at developmental disorders, I think in your population. So the contrast between your NAA levels and the choline levels kind of in different directions. And where we noticed in our sample, they're actually going in the same direction. So controls, TDC, typically developing controls, having higher levels of choline and NA compared to ADHD. So it is interesting to see in a healthy developmental population how you, I don't know if you had any thoughts about why you have these different contrasts.
Right. I agree. I think that's one of the reasons that it's important for us to establish these typical developmental trajectories and kind of norms of what should be expected in healthy development. I think with the changes that occur, it can also be very tissue dependent. So if you're sampling from a voxel that's more gray matter versus more white matter, you might see these effects differently. In all of my analyses, I've controlled for the tissue content of the voxels. Right. And right now we're collecting from some of these kids an additional like deep white matter voxel, because that's we've been missing that here. I hate to say it, but our voxel is kind of contaminated by gray matter for really understanding these specifically white matter tissue effects. So I think that's something to consider as well. If, in our data, we see that there's these patterns of, increasing NAA and decreasing choline, if that could just be driven differently in your data based on what part of the brain you're looking at and what tissues you're looking at.
Right. I believe it was also the ACC.
Okay. Yeah, which I like the ACC because it's mostly gray matter, and a lot of other studies have also looked at ACC. I know in there is a study from dyslexia that found also differences in the ACC with like processing speed and dyslexia. But again, it was the elevated choline that was associated with the poor outcomes. So I do wonder what factor may be related to the prenatal alcohol exposure could be driving potentially a way that choline is metabolized in those kids differently.
Interesting. Okay. I have a few other questions, but let the people ask them. And we have one question on chat, but before we go there, anyone else, please feel free to unmute yourself and ask. So meanwhile, Marco, so he's actually in Italy. He's joined on Zoom from there, and it's quite a big question. So Meaghan, do you want to actually read it and interpret it for us and then answer?
Sure, I will read it for those who may not be able to. So the question is that the field of TMS combined with MRS has been largely disappointing in the sense that TMS protocols show specific inhibitory or excitatory changes that predict specific MRS changes, yet those changes haven't been seen much. This led to the hypothesis that MRS is better at assessing local metabolites over longer temporal timescales. Obviously, the key question here, and especially for the kind of work that you nicely presented today, thank you, is how long is the temporal timescale? Can you speculate about that? Thanks. So this is something I've been grappling with a bit. So it depends on the metabolite, I would say first. So especially something like glutamate can change pretty rapidly. And there are some studies that look at, morning, evening, after sleep kind of changes in glutamate levels over time that fluctuate. And as well as, I would say that's the main one for like these kind of diurnal fluctuations. We looked mainly at annual measurements of these metabolites. So, we hope that it's kind of giving us a good picture of overall their change over time. But I'm interested in looking at shorter timescales and changes in kind of what's going on if we look at changes in metabolites and then in tissue structure subsequently as well, and kind of are there time lagged effects that might be observed? Like after you see a fluctuation in choline, do you see a fluctuation in myelin or white matter structure? And yeah, in terms of the TMS, I would also think that you'd want to consider GABA as well. So being that in many TMS studies, you're doing inhibition, you'd want to also look at whether there's changes in GABA. I haven't covered GABA here. We didn't do the J editing techniques to measure GABA in our sample. So I can't really speculate on that one too much. But yes, I would say these excitatory inhibitory changes do happen, on an overall timescale with development. And as we see again in our sample, we were capturing that a bit with the GLX signal, and then through adolescence, this decrease in GLX, which we're hoping is an indicator, or we think is an indicator of this inhibitory excitatory balance. But in terms of TMS, I think it's an interesting question. I haven't looked specifically at those effects in the TMS literature. So I hope that gets at your question a bit.
So continuing this a bit,
Sure.
especially, sorry, did anyone say something? Yeah, Marco, thumbs up. I think you answered his question.
Okay.
So especially considering neurostimulation. So I was also involved in another study with Catherine Nahr here, where she was looking at major depression and doing ECT in patients with MDD. And so we looked at GLX. You mentioned GLX. So we looked at GLX and NAA, and we did notice a decrease with treatment. And that treatment was over a duration of few weeks, rather, or maybe a month, less than a month. So this temporal timescale thing, we did notice a decrease there. in contrast, we did another study again with kids, cerebral palsy, and we had an exercise intervention for them. And then we also looked at MRS, and we looked at NAA in particular. The voxel was in ACC.
Yes.
And we did see acute changes in a matter of a month. And so, yeah, it's kind of interesting to me.
Yes, I think MRS can be very useful in that way to see, you're going to see a more rapid change in something like MRS and in these metabolite levels, likely in response to an intervention or a stimulation, than you will see in tissue structure, things that take a lot longer to develop and change, and likely a lot more repetition of that stimulation to change the underlying tissue structure. So I think in my work, we have yet to optimize kind of the right developmental timescale and how to link that adequately with kind of the timescale we expect for our white matter changes, for example. So that's, if anyone has ideas, that's something I've been thinking about as well.
Oh, interesting. Is there any work? Well, maybe so there's this other group in South Africa, Cape Town. I'm sure you know Kirsty Donald and her group, and I believe they collected MRS, but maybe 2-year-olds and further. I don't know. I wonder if there's MRS data on infants. So that time scale.
Yes, I know of, I think 2 papers, and I'm not sure if it's their group. I would guess it is from their group or some collaborators from Cape Town looking. They actually have one of the only longitudinal studies of MRS development in those kids.
So it is probably Ernesta Menkes. Yeah.
Yeah, so they had, I think, like 5, 7, and 9-year-olds in one of those studies, and they were able to look at some of the trajectories of these metabolites there, and our effects were quite consistent with what they had reported as well. I think they also looked at HIV exposed kids in the studies that I'm thinking of.
Okay.
Yes.
So any other questions? If not, so maybe Meaghan stay here for a few minutes, and then
Sure.
So the seminar's over, and I guess we'll see you guys in fall. So have a good summer, all of you.